Bioenergetics, a comprehensive overview for CSIR NET JRF Life Sciences. Bioenergetics is the study of the flow and transformation of energy in biological systems, focusing on how organisms manage energy to drive cellular processes essential for life. It involves understanding the biochemical processes that convert energy from one form to another, primarily through the oxidation of nutrients, and how this energy is harnessed to perform work at the molecular level. Bioenergetics is fundamental to various cellular processes, including metabolism, muscle contraction, active transport, and biosynthesis. 1. Energy currency of cells, ATP, adenosine triphosphate. ATP is the central molecule in bioenergetics, serving as the primary energy carrier in cells. It consists of adenine, ribose, and 3 phosphate groups. The energy stored in the high energy bonds between the phosphate groups is released when ATP is hydrolyzed to ADP, adenosine diphosphate, and inorganic phosphate. The hydrolysis of ATP is coupled with many cellular processes, providing the necessary energy for functions like muscle contraction, protein synthesis, and ion transport across membranes. Important concept. ATP hydrolysis releases approximately 30.5 kilojoules per mole of energy under standard conditions, which is utilized in various biochemical reactions. 2. Glycolysis, anaerobic breakdown of glucose. Glycolysis is the first step in both aerobic and anaerobic respiration and occurs in the cytoplasm. It involves the breakdown of one molecule of glucose C6H12O6 into 2 molecules of pyruvate, releasing a small amount of energy in the form of ATP and NADH. Glycolysis consists of 10 enzyme catalyzed reactions, divided into 2 phases. The energy investment phase requires ATP, and the energy payoff phase produces ATP and NADH. The overall reaction of glycolysis is glucose plus $2 million plus 2 ATP right pointing arrow 2 pyruvate plus 4 ATP plus 2 NADH. 3. Aerobic respiration, the complete breakdown of glucose. Aerobic respiration takes place in the presence of oxygen and involves the complete oxidation of glucose to produce carbon dioxide, water, and a large amount of ATP. It consists of 3 major stages. Glycolysis, as already described, this occurs in the cytoplasm and produces pyruvate. Pyruvate decarboxylation link reaction pyruvate is transported into the mitochondria, where it is converted into acetyl-CoA, releasing CO2 and producing NADH. Citric acid cycle, Krebs cycle, acetyl-CoA enters the citric acid cycle, a series of reactions that occur in the mitochondrial matrix. Each turn of the cycle generates 3 NADH, 1 FADH2, and 1 ATP or GTP by substrate level phosphorylation, while releasing CO2 as a waste product. Electron transport chain, etc. The not an FADH2 produced in previous steps donate electrons to the electron transport chain in the inner mitochondrial membrane. This leads to the generation of a proton gradient across the membrane, which drives ATP synthesis through oxidative phosphorylation. Oxygen acts as the final electron acceptor, forming water. Total ATP yield. For each molecule of glucose, the complete aerobic respiration process yields approximately 38 ATP molecules. 4. Anaerobic respiration and fermentation. When oxygen is not available, cells revert to anaerobic respiration or fermentation. In the absence of oxygen, glycolysis is followed by fermentation to regenerate NAD+ and ensure continued ATP production. There are 2 main types of fermentation. Lactic acid fermentation. In muscles, when oxygen is limited, e.g., during intense exercise, pyruvate is reduced to lactic acid to regenerate NAD+. This occurs in human cells and some bacteria. The equation is pyruvate plus NADH right pointing arrow lactic acid plus NAD. Alcoholic fermentation. In yeast and some plant cells, pyruvate is converted into ethanol and CO2, regenerating NAD+ for glycolysis. The reaction is pyruvate right pointing arrow ethanol plus CO2. Both processes yield 2 ATP per glucose molecule, much less than aerobic respiration. 5. Chemiosmotic theory of ATP synthesis. The chemiosmotic theory, proposed by Peter Mitchell, explains how the proton gradient across the mitochondrial membrane drives ATP synthesis. During the electron transport chain, protons H+ are pumped across the inner mitochondrial membrane into the intermembrane space, creating an electrochemical gradient. Protons flow back into the matrix through the ATP synthase enzyme complex, driving the conversion of ADP and inorganic phosphate into ATP. This process is known as oxidative phosphorylation. 6. Energy yield in bioenergetics. The efficiency of cellular energy production varies between different types of metabolism. For example, in aerobic respiration, the energy yield is much higher, around 38 ATP molecules per glucose compared to anaerobic processes, 2 ATP per glucose. Additionally, factors such as substrate availability, enzyme activity, and oxygen levels influence the overall efficiency of bioenergetic processes. Previous year questions with answers. Question 1, 2018. Which of the following molecules is directly involved in the formation of ATP during oxidative phosphorylation? A. PAD. B. NADHC, oxygen D. ATP synthase. Answer: D. ATP synthase. Explanation: ATP synthase is the enzyme responsible for synthesizing ATP during oxidative phosphorylation by utilizing the proton gradient established by the electron transport chain. Question 2, 2020. Which step of aerobic respiration directly generates ATP by substrate level phosphorylation? A. Glycolysis. B. Citric acid cycle. C. Electron transport chain. D. Pyruvate decarboxylation. Answer: B. Citric acid cycle. Explanation: the citric acid cycle generates ATP through substrate level phosphorylation, particularly in the conversion of succinyl-CoA to succinate. Question 3, 2017. The primary function of oxygen in cellular respiration is to act as a donor of electrons. B. Act as a final electron acceptor in the electron transport chain. C. Generate ATP by substrate level phosphorylation. D. Directly participate in the citric acid cycle. Answer: B. Act as a final electron acceptor in the electron transport chain. Explanation: oxygen accepts electrons and combines with protons to form water, completing the electron transport chain and allowing continued ATP production. This lecture provides a comprehensive foundation of bioenergetics, focusing on key concepts such as ATP, glycolysis, aerobic respiration, and the role of oxygen in cellular energy production. The questions included are aligned with CSIR NET Life Sciences topics, ensuring relevant and exam-focused preparation. You said, glycolysis, ChatGPT said. Glycolysis, detailed lecture for CSIR NET JRF Life Sciences. Glycolysis is a critical metabolic pathway that occurs in the cytoplasm of both prokaryotic and eukaryotic cells. It is the process through which glucose, a 6-carbon sugar, is broken down into 2 molecules of pyruvate, a 3-carbon compound producing ATP and NADH in the process. Glycolysis serves as the initial pathway for both aerobic and anaerobic respiration, making it a central biochemical pathway for energy production in all living organisms. This process does not require oxygen, making it essential in both oxygen-rich and oxygen-limited environments. 1. Overview of glycolysis. Glycolysis is a series of 10 enzyme catalyzed reactions that convert glucose into pyruvate. The pathway is divided into 2 phases: the energy investment phase and the energy payoff phase. Energy investment phase. In the first 5 steps, energy is required to initiate the process. 2 ATP molecules are consumed to phosphorylate glucose and its intermediates, preparing them for further breakdown. Energy payoff phase. In the next 5 steps, energy is released as ATP and NADH are produced. This phase results in the formation of 2 molecules of pyruvate. The overall reaction of glycolysis is Glucose C6H12O6 plus $2 million plus 2 ATP right pointing arrow 2 pyruvate C3H4O3 plus 4 ATP plus 2 NADH plus 2 HO. Thus, glycolysis is an anaerobic process that produces a net gain of 2 ATP and 2 NADH per molecule of glucose. 2. Detailed steps of glycolysis. The 10 steps of glycolysis are catalyzed by specific enzymes, each serving a distinct purpose. Step 1. Phosphorylation of glucose enzyme, hexokinase or glucokinase in liver cells. Reaction. Glucose is phosphorylated to form glucose 6-phosphate G6P using 1 ATP molecule. This step is important as it traps glucose within the cell and keeps it from leaving. ATP used. 1. Step 2. Isomerization of G6P enzyme, phosphoglucose isomerase. Reaction. Glucose 6-phosphate is isomerized to fructose 6-phosphate F6P. This is a simple rearrangement of atoms without any ATP usage. Step 3. Phosphorylation of F6P enzyme, PHOS-PHOFRUCTOKINASE1-PFK1. Reaction. Fructose 6-phosphate is phosphorylated to form fructose 1,6-phosphate F1, 6-BP using 1 ATP molecule. This step is a major regulatory point in glycolysis. PFK1 is allosterically regulated by various molecules like ATP inhibitor and AMP activator. ATP used. 1. Step 4. Cleavage of F1, 6-BP enzyme, aldolase. Reaction. The 6-carbon molecule fructose 1, 6-phosphate is split into 2 3-carbon molecules, dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate G3P. Only G3P continues through the pathway, but DHAP is isomerized into G3P. Step 5. Interconversion of DHAP to G3P enzyme, triphosphate isomerase. Reaction. Dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. After this step, there are 2 molecules of G3P that will proceed through the rest of glycolysis. Step 6. Oxidation of G3P. Enzyme, glyceraldehyde 3-phosphate dehydrogenase, GAPDH. Reaction. Each G3P molecule is oxidized, reducing NAD to NADH and adding an inorganic phosphate to form 1,3-bisphosphoglycerate 1,3-BPG. This is the first energy-producing step of glycolysis, as NADH is generated. NADH produced, 2,1 per G3P. Step 7. ATP generation, first ATP production, enzyme, phosphoglycerate kinase. Reaction. 1,3-bisphosphoglycerate donates a phosphate group to ADP, producing ATP and 3-phosphoglycerate 3-PG. Reaction. 1,3-bisphosphoglycerate donates a phosphate group to ADP, producing ATP and 3-phosphoglycerate 3-PG. This is the first substrate level phosphorylation step in glycolysis. ATP produced, 2,1 per G3P. Step 8. Isomerization of 3-PG enzyme. This is the first substrate level phosphorylation step in glycolysis. ATP produced, 2,1 per G3P. Step 8. Isomerization of 3-PG enzyme, phosphoglycerate mutase. Reaction. 3-phosphoglycerate is converted into 2-phosphoglycerate 2-PG. This is a simple rearrangement with no ATP production. Step 9. Dehydration of 2-PG enzyme, enolase. Reaction. 2-phosphoglycerate undergoes dehydration to form phosphoenolpyruvate, PEP, a high-energy compound. Water is released in this reaction. Step 10. ATP generation, second ATP production, enzyme, pyruvate kinase. Reaction. Phosphoenolpyruvate donates a phosphate group to ADP, producing ATP and pyruvate. This is the second substrate level phosphorylation step in glycolysis. ATP produced, 2,1 per G3P. At the end of glycolysis, the net result is the production of 2 ATP molecules, 2 NADH molecules, and 2 pyruvate molecules for each glucose molecule metabolized. 3. Regulation of glycolysis. Glycolysis is tightly regulated to meet the cell's energy needs. The key regulatory steps are hexokinase, glucokinase. These enzymes are inhibited by the product, glucose 6-phosphate. In the liver, glucokinase is regulated by the availability of glucose. PFK1. This is the main rate-limiting enzyme in glycolysis. It is allosterically inhibited by ATP and activated by AMP, reflecting the cell's energy status. Pyruvate kinase. Pyruvate kinase is regulated by phosphorylation and is inhibited by ATP and acetyl-CoA. 4. Fate of pyruvate. At the end of glycolysis, the pyruvate form can have several fates depending on the availability of oxygen. Aerobic conditions. Pyruvate enters the mitochondria and is converted into acetyl-CoA, which enters the citric acid cycle for further oxidation and aerobic respiration. Anaerobic conditions. Pyruvate is converted to lactate in animals, lactic acid fermentation, or ethanol and CO2 in yeast, alcoholic fermentation, to regenerate NAD+ and allow glycolysis to continue. Previous year questions with answers. Question 1, 2019. Which of the following is a direct product of the glycolytic pathway? A. NOD. B. FADH2C, acetyl-CoA D, oxygen. Answer. A. NOD. Explanation. NADH is produced in step 6 of glycolysis, during the oxidation of glyceraldehyde 3-phosphate. Question 2, 2017. The phosphorylation of glucose to glucose 6-phosphate in the first step of glycolysis is catalyzed by A. Hexokinase. B. Phosphofructokinase C, pyruvate kinase. D. Glyceraldehyde 3-phosphate dehydrogenase. Answer. A. Hexokinase. Explanation. Hexokinase or glucokinase in the liver catalyzes the phosphorylation of glucose to glucose 6-phosphate, using ATP. Question 3, 2020. In glycolysis, ATP is synthesized by a substrate level phosphorylation B, oxidative phosphorylation C, photophosphorylation D, chemiosmosis. Answer. A substrate level phosphorylation. Explanation. ATP is generated by substrate level phosphorylation in glycolysis, particularly during steps 7 and 10. This lecture provides a comprehensive overview of glycolysis, emphasizing its key steps, regulation, and relevance in both aerobic and anaerobic conditions. The included questions ensure a focus on the critical elements that are most likely to appear in the CSIR Net Life Sciences exam. You said. Oxidative phosphorylation. ChatGPT said. Oxidative phosphorylation. Detailed lecture for CSIR Net JRF Life Sciences. Oxidative phosphorylation is the final and most energy-efficient step of cellular respiration, occurring in the mitochondria of eukaryotic cells. It involves the transfer of electrons through the electron transport chain, etc., and the synthesis of ATP via the enzyme ATP synthase, driven by the electrochemical gradient of protons H+ across the mitochondrial membrane. This process is coupled with the oxidation of nutrients, primarily NADH and FADH2, and the reduction of molecular oxygen to form water. Oxidative phosphorylation is a critical process for generating the majority of ATP required by cells for energy-demanding activities such as muscle contraction, protein synthesis, and oxidative phosphorylation is a critical process for generating the majority of ATP required by cells for energy-demanding activities such as muscle contraction, protein synthesis, and active transport. 1. Structure of mitochondria and the location of oxidative phosphorylation. The mitochondrion is a double-membrane organelle. The outer membrane is permeable to small molecules, while the inner membrane is highly selective and contains the proteins necessary for the electron transport chain and ATP synthesis. Inner mitochondrial membrane contains the components of the electron transport chain, etc., and ATP synthase. Oxidative phosphorylation is a critical process for generating the majority of ATP required by cells for energy-demanding activities such as muscle contraction, protein synthesis, and active transport. 1. Structure of mitochondria and the location of oxidative phosphorylation. The mitochondrion is a double-membrane organelle. The outer membrane is permeable to small molecules, while the inner membrane is highly selective and contains the proteins necessary for the electron transport chain and ATP synthesis. Inner mitochondrial membrane contains the components of the electron transport chain, etc., and ATP synthase. Matrix, the inner compartment of the mitochondrion, where the citric acid cycle, Krebs cycle occurs and where NADH and FADH2 are generated. Intermembrane space, the region between the inner and outer membranes where protons H+ are pumped during the electron transport chain. 2. The electron transport chain, ETC. The electron transport chain is composed of a series of protein complexes located in the inner mitochondrial membrane. These complexes, complex I, IV, facilitate the transfer of electrons from NADH and FADH2 to molecular oxygen, the final electron acceptor. Complex I, NADH dehydrogenase, NADH donates electrons to complex I, which then transfers the electrons to ubiquinone Q. In the process, protons are pumped across the inner mitochondrial membrane from the matrix into the intermembrane space. NADH right pointing arrow NAD plus 2E plus 2H. Complex 2 succinate dehydrogenase, FADH2 donates electrons to complex 2, which passes them to ubiquinone, but does not pump protons. FADH2 right pointing arrow CAD plus 2E plus 2H. Ubiquinone Q, ubiquinone, also called coenzyme Q, is a lipid-soluble electron carrier that transfers electrons from complex I and complex 2 to complex 3. Complex 3 cytochrome B1 complex. Complex 3 transfers electrons from ubiquinone to cytochrome C and pumps protons into the intermembrane space. QH right pointing arrow Q plus 2B plus 2H. Cytochrome C. Cytochrome C is a small heme containing protein that carries electrons from complex 3 to complex IB. Complex IB, cytochrome C oxidase. Complex IB transfers electrons from cytochrome C to molecular oxygen, which is reduced to form water. In this process, additional protons are pumped into the intermembrane space. 4E plus 4H plus O right pointing arrow 2HO. 3. Proton gradient and chemiosmosis. As electrons move through the electron transport chain, protons H are actively pumped from the mitochondrial matrix into the intermembrane space by complexes I, 3, and IB. This creates an electrochemical gradient, also known as the proton motive force (PMF), across the inner mitochondrial membrane. The proton gradient results in a higher concentration of protons in the intermembrane space compared to the matrix. The electrochemical gradient, combined concentration and electrical gradients, stores potential energy, similar to water behind a dam. 4. ATP synthesis by ATP synthase. ATP synthase is a membrane-bound enzyme that utilizes the proton gradient created by the etc. to synthesize ATP. As protons flow back into the mitochondrial matrix through ATP synthase, their movement drives the phosphorylation of ADP to ATP in a process known as chemiosmosis. F subunit, the proton channel of ATP synthase, allowing protons to flow down their electrochemical gradient from the intermembrane space to the matrix. F subunit, the catalytic portion of ATP synthase, where ATP is synthesized from ADP and inorganic phosphate pi using the energy provided by the proton flow. For every 3 protons that flow through ATP synthase, 1 molecule of ATP is synthesized. 5. Oxygen is the final electron acceptor. Molecular oxygen plays a crucial role in oxidative phosphorylation by acting as the final electron acceptor in the electron transport chain. Without oxygen, the electron transport chain cannot function, as the system would become backed up, and NADH and FADH2 would not be oxidized. The reduction of oxygen to water at complex IB is essential to maintaining the flow of electrons and sustaining ATP production. 6. Energy yield from oxidative phosphorylation. The primary function of oxidative phosphorylation is to produce ATP, the energy currency of the cell. The amount of ATP produced depends on the number of electrons donated by NADH and FADH2. NADH, each NADH molecule produced in the citric acid cycle or glycolysis can yield approximately 2.5 ATP molecules through oxidative phosphorylation. FADH2, each FADH2 molecule produces approximately 1.5 ATP molecules. In total, the complete oxidation of one molecule of glucose via glycolysis, the citric acid cycle, and oxidative phosphorylation can produce a theoretical maximum of 38 ATP molecules. 7. Inhibitors and uncouplers of oxidative phosphorylation. Various substances can affect the efficiency of oxidative phosphorylation. Inhibitors. These compounds inhibit specific complexes of the electron transport chain or ATP synthase, blocking ATP production. For example, cyanide inhibits complex IB by binding to cytochrome C oxidase. Rhodinone inhibits complex I by preventing the transfer of electrons from NADH to ubiquinone. Uncouplers. Uncoupling agents disrupt the proton gradient across the inner mitochondrial membrane without allowing protons to pass through ATP synthase, leading to heat generation instead of ATP synthesis. Dinitrophenol DNP is an example of an uncoupler. Previous year questions with answers. Question 1, 2018. Which of the following is a consequence of oxygen being the final electron acceptor in the electron transport chain? A. ATP is generated by substrate level phosphorylation. B. The proton gradient across the inner mitochondrial membrane is maintained. C. NADH is oxidized in the cytoplasm. D. The electron transport chain becomes inhibited. Answer. B. The proton gradient across the inner mitochondrial membrane is maintained. Explanation. Oxygen acts as the final electron acceptor in the etc., allowing the continued transfer of electrons, which maintains the proton gradient across the inner mitochondrial membrane, essential for ATP production. Question 2, 2019. Which complex in the electron transport chain is directly involved in the reduction of oxygen to water? A. Complex I. B. Complex 2C. Complex 3D. Complex IB. Answer. D. Complex IB. Explanation. Complex IB, also known as cytochrome C oxidase, is responsible for the final reduction of oxygen to water in the electron transport chain. Question 3, 2017. The proton motive force PMF that drives ATP synthesis is generated by the flow of protons through ATP synthase B, the electrochemical gradient of protons created by the electron transport chain C, the reduction of oxygen in complex IB. D. The hydrolysis of ATP. Answer. B. The electrochemical gradient of protons created by the electron transport chain. Explanation. The proton motive force is created by the electron transport chain, which pumps protons into the intermembrane space, generating an electrochemical gradient that drives ATP synthesis via ATP synthase. This detailed lecture on oxidative phosphorylation outlines its key processes, structures involved, and critical steps in ATP production, ensuring thorough preparation for the CSIR Net Life Sciences exam. The included questions focus on core concepts and likely exam topics. You said, coupled reaction, ChatGPT said. Coupled reactions, detailed lecture for CSIR Net JRF Life Sciences. In biochemistry, coupled reactions refer to the process where an exergonic energy-releasing reaction is paired with an endergonic energy-consuming reaction. This coupling allows cells to perform essential tasks that require energy input, such as the synthesis of molecules or the transport of ions across membranes, by utilizing the energy released from other biochemical processes. Coupled reactions are fundamental to cellular metabolism and are most commonly seen in processes like ATP hydrolysis, oxidation reduction reactions, and biosynthesis. The principle behind coupled reactions is that the energy released from one reaction can be directly used to drive another reaction that would otherwise be non-spontaneous. 1. ATP hydrolysis is a common coupling mechanism. A classic example of a coupled reaction in biological systems is the use of ATP hydrolysis to power various biochemical processes. The hydrolysis of ATP, the breakdown of ATP into ADP and inorganic phosphate, high releases a significant amount of energy, which can be used to drive endergonic reactions. ATP right pointing arrow ADP plus high plus energy. This exergonic reaction releases about 7.3 kilocalories per mole of energy, which is utilized to drive other metabolic processes such as muscle contraction, active transport, and the synthesis of macromolecules like proteins and nucleic acids. Coupling ATP hydrolysis to an endergonic reaction enables cells to perform necessary tasks that would not otherwise occur spontaneously due to the positive Gibbs free energy delta G. These coupled reactions can be seen in various cellular processes. Muscle contraction. The hydrolysis of ATP by the motor protein myosin is coupled to the sliding of actin and myosin filaments during muscle contraction. Protein synthesis. The formation of peptide bonds between amino acids is an energy-consuming process, but ATP hydrolysis provides the energy to drive this reaction. 2. Oxidation reduction reactions is coupled reactions. Another critical example of coupled reactions occurs in redox reactions, oxidation reduction reactions, where the transfer of electrons from one molecule oxidation is directly coupled to the reduction of another molecule. This coupling plays a key role in cellular respiration and other metabolic pathways. For example, in the electron transport chain, etc., in oxidative phosphorylation, the transfer of electrons from NADH and FADH to the oxygen is coupled to the pumping of protons H+ across the inner mitochondrial membrane, creating a proton gradient. The energy stored in this gradient is then used to produce ATP via ATP synthase. NADH plus H plus 1/2 right pointing arrow NAD plus HO, oxidation of NADH and reduction of oxygen. Proton gradient formation. The energy released from the oxidation of NADH is coupled to the active transport of protons across the mitochondrial membrane. This is an example of a coupled redox reaction, where energy is transferred from one molecule to another through the movement of electrons, and the energy is harnessed to do useful work, such as the synthesis of ATP. 3. Coupled transport. Coupled reactions also play a role in the transport of molecules across membranes, especially when cells need to move substances against their concentration gradients. In these cases, the movement of one molecule down its concentration gradient provides the energy needed to transport another molecule against its gradient. This is called secondary active transport. Symporters. These transporters move two molecules in the same direction across a membrane. For example, the sodium-glucose transporter couples the movement of sodium ions, not down their electrochemical gradient with the movement of glucose against its concentration gradient. Now plus glucose outside, right arrow now plus glucose inside. Antiporters. These transporters move two molecules in opposite directions. For example, the sodium-potassium pump, not K-ATPase, uses ATP hydrolysis to pump now out of the cell and K into the cell, both against their concentration gradients. ATP right pointing arrow ADP plus high plus energy used to transport ions. This form of coupling enables cells to import necessary nutrients and export waste products without expending direct energy for each molecule by using the energy stored in ion gradients. 4. Coupling in biosynthetic pathways. Biosynthesis of macromolecules, such as proteins, nucleic acids, and lipids, is also powered by coupled reactions. These processes often require energy input, and ATP or other high-energy molecules are hydrolyzed to provide the necessary energy. For example, in protein synthesis, amino acids are linked together through peptide bonds in a process that requires energy. The reaction is coupled with the hydrolysis of ATP during the activation of amino acids and GTP during translation to drive the formation of proteins. Amino acid activation. ATP is used to attach an amino acid to its corresponding terna. Peptide bond formation. The ribosome catalyzes the formation of peptide bonds during translation, and GTP is used to drive the elongation cycle. 5. Gibbs free energy and coupling. Coupled reactions are primarily driven by Gibbs free energy, delta G. A reaction that releases energy, negative delta G, can drive a reaction that requires energy, positive delta G. The sum of the delta G values of the coupled reactions should be negative for the overall process to be spontaneous. Exergonic reaction. Delta G is negative, indicating that the reaction releases energy. Endergonic reaction. Delta G is positive, indicating that the reaction requires energy input. The coupling of an exergonic reaction, such as ATP hydrolysis, with an endergonic reaction, such as protein synthesis, allows cells to perform work that would otherwise not occur spontaneously. Previous year questions with answers. Question 1, 2019. Which of the following is true about coupled reactions in metabolism? A. The products of an exergonic reaction are always reactants in an endergonic reaction. B. Energy from ATP hydrolysis can be used to drive both exergonic and endergonic reactions. C. ATP synthesis is always exergonic and does not require coupling. D. NADH and FADH2 are never involved in coupled reactions. Answer. B. Energy from ATP hydrolysis can be used to drive both exergonic and endergonic reactions. Explanation. ATP hydrolysis is a key exergonic reaction in metabolism that can provide energy to drive endergonic reactions like biosynthesis, active transport, and muscle contraction. Question 2, 2017. Which of the following is an example of a coupled reaction? A. ATP hydrolysis used to drive muscle contraction. B. Glucose breakdown in the cytoplasm. C. Photosynthesis in plants. D. Oxygen consumption by mitochondria. Answer. ATP hydrolysis used to drive muscle contraction. Explanation. Muscle contraction is an endergonic process that is powered by the exergonic hydrolysis of ATP, making it an example of a coupled reaction. Question 3, 2018. In the electron transport chain, which of the following couples oxidation with the transport of protons across the membrane? A. Cytochrome C. B. ATP synthase. C. NADH dehydrogenase. D. Oxygen. Answer. C. NADH dehydrogenase. Explanation. NADH dehydrogenase complex I couples the oxidation of NADH with the transport of protons across the mitochondrial membrane, contributing to the creation of a proton gradient. This detailed lecture on coupled reactions provides a clear understanding of how cellular processes harness energy through coupling, essential for exam preparation. The included questions reflect key concepts and are aligned with CSIR Net Life Sciences exam expectations. You said. Group transfer. ChatGPT said. Group transfer. Detailed lecture for CSIR Net JRF Life Sciences. Group transfer reactions are biochemical processes where a functional group is transferred from one molecule, the donor, to another molecule, the acceptor. These reactions are central to many biochemical pathways, including metabolism, signal transduction, and macromolecule synthesis. The transfer of functional groups is often coupled with energy changes, and enzymes that catalyze these reactions are known as transferases. Group transfer is an essential mechanism in the regulation of cellular processes. It involves a variety of molecular groups such as phosphate groups, methyl groups, acetyl groups, amino groups, and others, depending on the specific biochemical pathway. These transferred groups can alter the properties of the recipient molecule, enabling the cell to regulate functions like enzyme activity, gene expression, and metabolic rates. 1. Mechanisms of group transfer. The transfer of a group from one molecule to another typically involves the following steps. Nucleophilic attack. The donor molecule has a functional group that is activated by a nucleophilic attack by the acceptor molecule. This results in the breaking of the bond between the donor and the transferred group. Formation of a new bond. The transfer forms a new covalent bond with the acceptor molecule, completing the transfer process. Enzyme catalysis. Most group transfer reactions are catalyzed by enzymes, which help stabilize the transition state and lower the activation energy required for the reaction to occur. 2. Common types of group transfer. Group transfer reactions occur in a variety of metabolic pathways, and different types of groups are transferred depending on the biochemical context. Below are some common examples. Phosphate group transfer. The transfer of a phosphate group, often from ATP, is one of the most common and important group transfers in metabolism. The enzymes responsible for these reactions are called kinases and phosphatases. For example, in glycolysis, the enzyme hexokinase transfers a phosphate group. For example, in glycolysis, the enzyme hexokinase transfers a phosphate group from ATP to glucose, forming glucose 6-phosphate. Another example is the phosphorylation of proteins, where kinases add phosphate groups to specific amino acids in proteins, influencing their activity and function. Acetyl group transfer. The transfer of acetyl groups is another vital reaction, especially in the regulation of gene expression and energy production. Acetyl-CoA serves as the primary donor of acetyl groups in various metabolic reactions. An example is the transfer of an acetyl group from acetyl-CoA to oxaloacetate to form citrate in the citric acid cycle, Krebs cycle. Additionally, acetylation of histone proteins regulates gene expression by modifying chromatin structure. Methyl group transfer. Methylation is a process by which a methyl group, CH, is transferred from a donor molecule such as S-adenosylmethionine or SAM to a substrate. This process is important in regulating gene expression, protein function, and signal transduction. DNA methylation is a key example, where a methyl group is added to the 5' position of cytosine residues in DNA, often silencing gene expression. Methylation of histones is another key regulatory mechanism in chromatin remodeling and gene expression. Amino group transfer. The transfer of an amino group, NH, is crucial for amino acid metabolism and the synthesis of nitrogen-containing compounds. This process is often catalyzed by enzymes called aminotransferases or transaminases. An example is the conversion of glutamate to alpha-ketoglutarate through the transfer of an amino group, a key step in nitrogen metabolism. 3. Role of coenzymes in group transfer. Many group transfer reactions require coenzymes to act as intermediates, facilitating the transfer of functional groups between donor and acceptor molecules. Some important coenzymes involved in group transfer reactions include ATP. ATP is the most common energy source for transferring phosphate groups. It donates phosphate groups to various substrates in reactions catalyzed by kinases and other enzymes. Coenzyme A-CoA. CoA carries acetyl groups and other acyl groups in cellular metabolism. Acetyl-CoA, for instance, donates acetyl groups in reactions like the citric acid cycle. S-adenosylmethionine, SAM. SAM is the primary methyl group donor in methylation reactions. It plays a crucial role in DNA methylation, protein methylation, and other methylation processes. Pyridoxal phosphate, PLP. PLP, the active form of vitamin B6, is a coenzyme involved in transamination reactions, where an amino group is transferred between amino acids. 4. Biological significance of group transfer. Group transfer reactions are essential for regulating various biological processes. Metabolic pathways. Group transfer is central to metabolic reactions such as glycolysis, the citric acid cycle, and fatty acid metabolism, where groups like phosphate, acetyl, and amino groups are transferred to generate energy or build complex molecules. Signal transduction. The transfer of phosphate groups via kinases is a key mechanism in signal transduction pathways. Phosphorylation often activates or deactivates enzymes or other proteins, thus regulating cellular responses to external signals. Gene regulation. Acetylation and methylation of DNA and histones are crucial for regulating gene expression. These modifications alter chromatin structure and accessibility, thus controlling whether a gene is transcribed or silenced. 5. Examples of group transfer reactions. Glycolysis. In the first step of glycolysis, the enzyme hexokinase transfers a phosphate group from ATP to glucose, forming glucose 6-phosphate. Citric acid cycle. Acetyl-CoA donates an acetyl group to oxaloacetate, forming citrate. This reaction is catalyzed by the enzyme citrate synthase. Amino acid metabolism. In the transamination of glutamate, the amino group is transferred to oxaloacetate to form aspartate, catalyzed by the enzyme glutamate oxaloacetate transaminase GOT. 6. Gives free energy and coupling in group transfer. Many group transfer reactions are coupled with energy-releasing reactions to ensure that the overall process is thermodynamically favorable. For example, the hydrolysis of ATP provides the energy needed for the phosphorylation of glucose in glycolysis. Previous year questions with answers. Question 1, 2018. Which of the following enzymes catalyzes the transfer of a phosphate group to glucose in glycolysis? A. Phosphofructokinase. B. Hexokinase. C. Glucose 6-phosphatase D. Pyruvate kinase. Answer. B. Hexokinase. Explanation. Hexase transfers a phosphate group from ATP to glucose, forming glucose 6-phosphate in the first step of glycolysis. Question 2, 2017. Which of the following enzymes is primarily involved in the transfer of methyl groups in biological reactions? A. NAD+. B. S-adenosylmethionine, SAM. C. Coenzyme A. D. ATP. Answer. B. S-adenosylmethionine, SAM. Explanation. SAM is a major methyl group donor in various methylation reactions, such as DNA and protein methylation. Question 3, 2019. The transfer of an acetyl group from acetyl-CoA to oxaloacetate in the citric acid cycle is catalyzed by which enzyme? A. Citrate synthase. B. Acetyl-CoA synthetase. C. Pyruvate dehydrogenase. D. Isocitric dehydrogenase. Answer. Citrate synthase. Explanation. Citrate synthase catalyzes the transfer of an acetyl group from acetyl-CoA to oxaloacetate, forming citrate in the first step of the citric acid cycle. This lecture on group transfer reactions provides a comprehensive overview of the processes, enzymes, and biological significance of group transfer in cellular metabolism. The included questions help reinforce key concepts relevant to CSIR-NET Life Sciences exam preparation. You said. Biological energy transducers. ChatGPT said. Biological energy transducers. Detailed lecture for CSIR-NET JRF Life Sciences. Biological energy transducers are specialized molecules or systems in living organisms that convert one form of energy into another, usually in a manner that facilitates the functioning of biological processes. These transducers play an essential role in converting biochemical energy from nutrients into usable forms of energy, such as ATP, which is used to drive various cellular activities. These systems are crucial for energy metabolism, cellular signaling, and maintaining homeostasis in living cells. 1. Role of energy transducers in biological systems. Living organisms require energy to perform work, maintain cellular structures, and carry out vital biochemical reactions. Biological energy transducers enable the conversion of energy between different forms, such as light energy, chemical energy, and mechanical energy. These transducers are primarily enzymes, coenzymes, and protein complexes involved in metabolic pathways, cellular processes, and energy transfer mechanisms. 2. Types of biological energy transducers. There are several types of biological energy transducers that play distinct roles in different biological systems. The most notable types include A. ATP synthase. ATP synthase is a crucial energy transducer in biological systems, particularly in the mitochondria of eukaryotic cells and in the plasma membranes of some prokaryotes. ATP synthase is responsible for synthesizing ATP from ADP and inorganic phosphate using the energy derived from a proton gradient across membranes chemiosmosis. Mechanism. In the electron transport chain, etc., protons are pumped across the inner mitochondrial membrane, creating a proton gradient. This proton gradient drives the rotation of the ATP synthase enzyme complex, facilitating the conversion of ADP and pi into ATP. This process occurs during oxidative phosphorylation. Biological importance. ATP is the primary energy currency in cells, used for various processes such as protein synthesis, cell division, muscle contraction, and ion transport. B. Photosystems. PSI and PSII. In plants, algae, and some bacteria, photosystems PSI and PSII are key biological energy transducers in the process of photosynthesis. These complexes convert light energy into chemical energy, which is then used to produce ATP and NADPH, vital for the synthesis of organic molecules. Mechanism. When light is absorbed by the chlorophyll in the photosystems, electrons are excited to higher energy levels. The energy from the excited electrons is then transferred through a series of electron carriers in the electron transport chain, etc., leading to the production of ATP via chemiosmosis and the reduction of ADP plus to NADPH in the stroma. Biological importance. Photosynthesis is a process through which plants, algae, and certain bacteria convert light energy into chemical energy, which sustains life on Earth by producing organic molecules and oxygen. C. Mitochondrial proton gradient chemiosmosis. The mitochondrial proton gradient is an essential component of energy conversion in eukaryotic cells, especially in the mitochondria, where it plays a key role in ATP production. It is part of the process of oxidative phosphorylation. Mechanism. During the electron transport chain, etc., electrons are transferred through protein complexes embedded in the inner mitochondrial membrane. The energy released is used to pump protons H+ across the membrane, creating an electrochemical gradient. This proton gradient drives ATP synthesis via ATP synthase through the process of chemiosmosis. Biological importance. The proton gradient allows the cell to efficiently convert energy from nutrients like glucose into ATP, which is essential for cellular functions like muscle contraction, protein synthesis, and maintaining cellular ion balance. D. Ion pumps and channels. Proton pumps. Sodium-potassium pump. Ion pumps and channels, such as the sodium-potassium pump, NOT plus K plus ATPase, and proton pumps, are critical transducers that convert chemical energy into mechanical energy in the form of ion gradients. These ion gradients are essential for maintaining osmotic balance and for cellular processes such as signal transmission and muscle contraction. Mechanism. For example, the NOT plus K plus ATPase pump uses ATP to transport sodium ions, NOT plus, out of the cell and potassium ions K plus into the cell, creating a concentration gradient that is used for various cellular processes, including the transmission of nerve impulses. Biological importance. Ion gradients are essential for a variety of cellular functions, including nerve signal transmission, nutrient uptake, and the maintenance of cellular integrity. E. Adenylate cyclase AC and guanylate cyclase GC. Adenylate cyclase AC and guanylate cyclase GC are enzymes that convert ATP and GTP into cyclic AMP, CAMP, and cyclic GMP, cGMP, respectively. These cyclic nucleotides act as second messengers in many signal transduction pathways. Mechanism. The conversion of ATP to CAMP by adenylate cyclase and GTP to cGMP by guanylate cyclase is often triggered by external signals, such as hormones, that bind to receptor proteins on the cell surface. Biological importance. CAMP and cGMP regulate various cellular processes, including metabolic pathways, gene expression, and cell division. They are involved in the signal transduction pathways that mediate the cellular response to hormones like adrenaline and nitric oxide. F. Flavin-containing proteins. Flavoproteins. Flavoproteins are enzymes that contain flavin cofactors, such as FMN flavin mononucleotide or fat flavin adenine dinucleotide, which play a crucial role in electron transfer reactions during cellular respiration and photosynthesis. Mechanism. Flavoproteins are involved in oxidation reduction reactions. They accept and donate electrons in a manner similar to NAD+ and NADH, acting as intermediates in the electron transport chain, etc. Biological importance. These proteins are involved in various energy transduction processes in both the mitochondria, cellular respiration, and chloroplast photosynthesis, contributing to the synthesis of ATP. 3. Energy transduction in cellular metabolism. Energy transduction is vital for cellular metabolism. The various enzymes, cofactors, and transmembrane complexes discussed above serve as intermediaries in the conversion of chemical, light, and mechanical energy into usable forms, such as ATP. In cellular respiration, energy from glucose metabolism is transferred to ATP molecules through glycolysis, the citric acid cycle, and oxidative phosphorylation. In photosynthesis, light energy is converted into chemical energy, which is stored in the form of ATP and NADPH, used to synthesize glucose from carbon dioxide and water. 4. Applications and significance. The biological transduction of energy allows organisms to efficiently carry out cellular functions like movement, signal transmission, biosynthesis, and growth. Disruptions in these energy conversion systems can lead to diseases such as mitochondrial disorders, cancer, and metabolic syndrome. 5. Previous year questions with answers. Question 1, 2016. Which of the following complexes is responsible for the final production of ATP in the electron transport chain? A. Complex I. B. Complex 2C. Complex VD. Complex IB. Answer. C. Complex B. Explanation. Complex B, also known as ATP synthase, is responsible for the final production of ATP in the electron transport chain by utilizing the proton gradient. Question 2, 2017. The conversion of light energy into chemical energy in photosynthesis is primarily carried out by which of the following? A. NADH dehydrogenase. B. Photosystem ion 2C ATP synthase D. Acetyl-CoA. Answer. B. Photosystem ion 2. Explanation. Photosystem ion 2 or the complex is responsible for the conversion of light energy to chemical energy, which is stored in the form of ATP and NADPH during photosynthesis. Question 3, 2018. The primary function of the NOT plus K plus ATPase pump in cells is to A. Produce ATP. B. Generate proton gradients. C. Maintain ion gradients across membranes. D. Synthesize CAMP. Answer. C. Maintain ion gradients across membranes. Explanation. The NOT plus K plus ATPase pump helps maintain the sodium and potassium ion gradients across the plasma membrane, which is essential for functions like nerve impulse transmission and cellular homeostasis. This lecture on biological energy transducers highlights the core mechanisms and systems involved in the conversion of different forms of energy in biological systems, emphasizing their importance in cellular metabolism and energy production. The included questions help reinforce key concepts for CSIR-NET Life Sciences exam preparation. You said. Principles of catalysis. ChatGPT said. Principles of catalysis. Detailed lecture for CSIR-NET JRF Life Sciences. Catalysis is a process by which the rate of a chemical reaction is increased by the presence of a substance known as a catalyst. The catalyst participates in the reaction but is not consumed or permanently altered in the process. Catalysis is a crucial phenomenon in various biological processes, including metabolic pathways, enzyme function, and industrial applications. It plays an essential role in reducing the activation energy required for a reaction to proceed, thereby speeding up the reaction rate. 1. Definition of catalysis. Catalysis refers to the acceleration of a chemical reaction due to the presence of a substance called a catalyst. A catalyst works by providing an alternative reaction pathway with a lower activation energy. It is important to note that catalysts do not alter the equilibrium of the reaction or the products formed, they only speed up the reaction. 2. Types of catalysis. There are two main types of catalysis, homogeneous catalysis and heterogeneous catalysis. Both types can be either positive, accelerating reactions, or negative, inhibiting reactions. A. Homogeneous catalysis. In homogeneous catalysis, the catalyst exists in the same phase, usually liquid or gas, as the reactants. An example of homogeneous catalysis is the use of acids or bases to catalyze certain chemical reactions in solution. Mechanism. In this case, the catalyst interacts directly with the reactants, forming an intermediate complex that facilitates the conversion of reactants to products. Example, the acid catalyzed esterification reaction, where an acid like sulfuric acid HSO catalyzes the reaction between an alcohol and a carboxylic acid to form an ester. B. Heterogeneous catalysis. In heterogeneous catalysis, the catalyst is in a different, usually solid, than the reactants, which are often gases or liquids. This type of catalysis is commonly used in industrial processes. Mechanism. The reactants absorb onto the surface of the solid catalyst, where they undergo chemical transformations. The products are then desorted from the cysts. Example, the Haber process, used to synthesize ammonia from nitrogen and hydrogen gases, involves a solid iron catalyst. 3. Catalytic cycle and mechanism. The catalytic process typically follows a series of steps known as the catalytic cycle, which involves the following stages. Adsorption. Reactants are absorbed onto the surface of the catalyst, where they are brought closer together and oriented in a favorable manner for the reaction. Reaction. Once absorbed, the reactants undergo the chemical transformation, forming an intermediate complex. The catalyst may stabilize this intermediate, lowering the activation energy. Desorption. The product of the reaction is released from the catalyst surface. The catalyst is now free to catalyze additional reactions. This cycle repeats, allowing the catalyst to facilitate many reactions without being consumed. 4. Enzyme catalysis. Enzymes are biological catalysts that speed up biochemical reactions in living organisms. They are highly specific and efficient and function under mild conditions, temperature, pH, and pressure. The principles of enzyme catalysis include active site. Enzymes have a specific region known as the active site, where substrates bind and undergo catalysis. The active site's shape and chemical environment are crucial for the enzyme's function. Induced fit model. According to this model, the enzyme undergoes a conformational change when the substrate binds to its active site. This change helps position the substrate in an optimal orientation for the reaction. Lowering activation energy. Enzymes lower the activation energy of a reaction by providing a specific environment for the reaction to occur, often involving the formation of transient covalent bonds with the substrate. Example, the enzyme amylase catalyzes the hydrolysis of starch into sugar molecules, speeding up the breakdown of starch in the digestive system. 5. Factors affecting catalysis. Several factors can influence the rate of catalysis and the efficiency of catalysts, including A. Concentration of reactants. Higher concentrations of reactants typically lead to faster reactions because more molecules are available for collision with the catalyst. B. Temperature. Increasing temperature generally increases the rate of reaction, as particles move faster, leading to more frequent collisions with the catalyst. However, enzymes have an optimal temperature range, and temperatures above this range can lead to enzyme denaturation. C. pH. For enzyme catalysis, each enzyme has an optimal pH at which it is most active. Deviations from this optimal pH can lead to a decrease in catalytic efficiency due to changes in the enzyme structure. D. Catalyst concentration. In homogeneous catalysis, increasing the concentration of the catalyst can increase the reaction rate. However, in heterogeneous catalysis, this relationship is not always linear because the surface area of the catalyst can become saturated with reactants. E. Inhibitors and activators. Inhibitors decrease the catalytic activity, while activators it. Inhibition can be competitive, non-competitive, or incompetitive, depending on how the inhibitor interacts with the enzyme or catalyst. 6. Mechanism of enzyme catalysis. Enzyme catalysis occurs through several mechanisms that involve various chemical and physical interactions between the enzyme and its substrate. Homogeneous catalysis. In homogeneous catalysis, the catalyst exists in the same phase, usually liquid or gas, as the reactants. An example of homogeneous catalysis is the use of acids or bases to catalyze certain chemical reactions in solution. Mechanism. In this case, the catalyst interacts directly with the reactants, forming an intermediate complex that facilitates the conversion of reactants to products. Example, the acid-catalyzed esterification reaction, where an acid like sulfuric acid HSO catalyzes the reaction between an alcohol and a carboxylic acid to form an ester. B. Heterogeneous catalysis. In heterogeneous catalysis, the catalyst is in a different phase, usually solid, than the reactants, which are often gases or liquids. This type of catalysis is commonly used in industrial processes. Mechanism. The reactants absorb onto the surface of a solid catalyst, where they undergo chemical transformations. The products are then desorted from the surface. Example, the Haber process, used to synthesize ammonia from nitrogen and hydrogen gases, involves a solid iron catalyst. 3. Catalytic cycle and mechanism. The catalytic process typically follows a series of steps known as the catalytic cycle, which involves the following stages. Adsorption. Reactants are absorbed onto the surface of the catalyst, where they are brought closer together and oriented in a favorable manner for the reaction. Reaction. Once absorbed, the reactants undergo the chemical transformation, forming an intermediate complex. The catalyst may stabilize this intermediate, lowering the activation energy. Desorption. The product of the reaction is released from the catalyst surface. The catalyst is now free to catalyze additional reactions. This cycle repeats, allowing the catalyst to facilitate many reactions without being consumed. 4. Enzyme catalysis. Enzymes are biological catalysts that speed up biochemical reactions in living organisms. They are highly specific and efficient and function under mild conditions, temperature, pH, and pressure. The principles of enzyme catalysis include active site. Enzymes have a specific region known as the active site, where substrates bind and undergo catalysis. The active site's shape and chemical environment are crucial for the enzyme's function. Induced fit model. According to this model, the enzyme undergoes a conformational change when the substrate binds to its active site. This change helps position the substrate in an optimal orientation for the reaction. Lowering activation energy. Enzymes lower the activation energy of a reaction by providing a specific environment for the reaction to occur, often involving the formation of transient covalent bonds with the substrate. Example, the enzyme amylase catalyzes the hydrolysis of starch into sugar molecules, speeding up the breakdown of starch in the digestive system. 5. Factors affecting catalysis. Several factors can influence the rate of catalysis and the efficiency of catalysts, including A. Concentration of reactants. Higher concentrations of reactants typically lead to faster reactions because more molecules are available for collision with the catalyst. B. Temperature. Increasing temperature generally increases the rate of reaction, as particles move faster, leading to more frequent collisions with the catalyst. However, enzymes have an optimal temperature range, and temperatures above this range can lead to enzyme denaturation. C. pH. For enzyme catalysis, each enzyme has an optimal pH at which it is most active. Deviations from this optimal pH can lead to a decrease in catalytic efficiency due to changes in the enzyme structure. Catalyst concentration. In homogeneous catalysis, increasing the concentration of the catalyst can increase the reaction rate. However, in heterogeneous catalysis, this relationship is not always linear because the surface area of the catalyst can become saturated with reactants. E. Inhibitors and activators. Inhibitors decrease the catalytic activity, while activators enhance it. Inhibition can be competitive, non-competitive, or incompetitive, depending on how the inhibitor interacts with the enzyme or catalyst. 6. Mechanism of enzyme catalysis. Enzyme catalysis occurs through several mechanisms that involve various chemical and physical interactions between the enzyme and its substrate. Proximity and orientation effect. The enzyme brings the substrates into close proximity and aligns them in the correct orientation, increasing the likelihood of successful collisions. Transition state stabilization. Enzymes stabilize the transition state of the substrate, lowering the activation energy required for the reaction. Covalent catalysis. Some enzymes form temporary covalent bonds with the substrate during the reaction, helping to stabilize the reaction intermediate. Acid-base catalysis. Some enzymes use acidic or basic groups in their active sites to donate or accept protons, facilitating the reaction. Metal ion catalysis. Many enzymes require metal ions, e.g., Mg squared, Zn squared, Fe squared in their active sites to stabilize charged intermediates and participate in electron transfer. 7. Catalysis in industry and biotechnology. Catalysis has significant industrial applications, ranging from the production of chemicals and fuels to environmental protection. Catalytic converters. In automobile exhaust systems, catalysts like platinum, palladium, and rhodium are used to convert harmful gases such as carbon monoxide, hydrocarbons, and nitrogen oxides into less harmful products like carbon dioxide and nitrogen. Biocatalysis. Enzymes are increasingly used in industrial processes, e.g., the synthesis of pharmaceuticals, food processing due to their specificity, milder conditions, and fewer byproducts compared to traditional chemical catalysts. 8. Previous year questions with answers question 1, 2015. Which of the following statements is true about enzyme catalysis? A. Enzymes increase the activation energy of a reaction. B. Enzymes lower the activation energy of a reaction. C. Enzymes are consumed in a reaction. D. Enzymes are not specific to their substrates. Answer. B. Enzymes lower the activation energy reaction. Explanation. And speed up biochemical reactions by lowering the activation energy required for the reaction to proceed. Question 2, 2017. Which type of catalysis involves the catalyst and reactants being in the same phase? A. Heterogeneous catalysis. B. Homogeneous catalysis. C. Enzyme catalysis. D. Surface catalysis. Answer. B. Homogeneous catalysis. Explanation. In homogeneous catalysis, the catalyst and reactants are in the same phase, typically liquid or gas. Question 3, 2019. The mechanism of enzyme catalysis that involves the formation of a temporary covalent bond between the enzyme and substrate is called a proximity effect. B. Acid-base catalysis. C. Covalent catalysis. D. Metal ion catalysis. Answer. C. Covalent catalysis. Explanation. Covalent catalysis involves the formation of a transient covalent bond between the enzyme and the substrate, helping to stabilize the reaction intermediate. Conclusion. Understanding the principles of catalysis is fundamental to mastering biochemical processes in living organisms and industrial systems. Catalysts, particularly enzymes, lower the activation energy of reactions, facilitating numerous vital processes in biology and industry. By enhancing reaction rates without being consumed, catalysts are indispensable in maintaining cellular function and in many technological applications. You said. Enzymes and enzyme kinetics. ChatGPT said. Enzymes and enzyme kinetics. Detailed lecture for CSIR Net JRF Life Sciences. Enzymes are biological catalysts that accelerate chemical reactions in living organisms. They are highly specific, efficient, and play a crucial role in metabolic pathways. Enzymes can catalyze both anabolic building and catabolic breaking down reactions, and they function under mild conditions, such as body temperature and physiological pH. The study of enzymes and how they function, known as enzyme kinetics, is vital for understanding their role in biochemical processes. 1. Introduction to enzymes. Enzymes are proteins or sometimes RNA molecules, known as ribozymes, that accelerate biochemical reactions by lowering the activation energy required for the reactions to proceed. Enzymes work through their active sites, where substrates bind and undergo chemical transformations. These transformations result in the formation of products, which are then released. A. Basic features of enzymes. Specificity. Enzymes are highly specific in terms of the substrate they act on. This specificity is determined by the shape and chemical environment of the enzyme's active site. Regulation. Enzyme activity can be regulated by various factors such as temperature, pH, substrate concentration, and the presence of inhibitors or activators. Efficiency. Enzymes are incredibly efficient catalysts, accelerating reactions by millions of times compared to non-catalyzed reactions. B. Structure of enzymes. Enzymes are typically globular proteins with a well-defined three-dimensional structure. The enzyme's active site is the region where the substrate binds. The shape and properties of the active site are highly complementary to the substrate, ensuring specificity. Active site. This region of the enzyme binds the substrate and undergoes a conformational change to facilitate the reaction. The mechanism of this binding and transformation is often described by the induced fit model, where the enzyme's active site adjusts slightly to better fit the substrate upon binding. 2. Enzyme-substrate complex. The interaction between the enzyme and its substrate is a crucial step in enzyme catalysis. This interaction forms an enzyme-substrate S complex. Lock and key model. This model suggests that the enzyme's active site has a specific shape that exactly fits the substrate, similar to a lock and a key. Induced fit model. This more accepted model proposes that the enzyme's active site is flexible and can adjust its shape upon substrate binding, enhancing the fit. Once the S complex is formed, the enzyme catalyzes the conversion of the substrate into the product. The product is released from the active site, and the enzyme is free to catalyze further reactions. 3. Enzyme kinetics. Enzyme kinetics is the study of the rates of enzyme catalyzed reactions. It focuses on how factors such as substrate concentration, enzyme concentration, temperature, and pH affect the rate of the reaction. Key concepts in enzyme kinetics include: A. Michaelis-Menten kinetics. The Michaelis-Menten model describes the relationship between enzyme activity and substrate concentration. It assumes that the enzyme forms a reversible complex with the substrate, and the reaction follows a simple two-step mechanism. Formation of the enzyme-substrate complex S. Conversion of the complex to product and enzyme release. The rate of the reaction B is dependent on the concentration of the substrate, and this relationship is described by the Michaelis-Menten equation. B equals B max S Km plus S versus equals Km. Plus S B. Max. S. Where B is the rate of the reaction. B underscore max is the maximum rate when the enzyme is saturated with substrate. S is the concentration of the substrate. K underscore M is the Michaelis constant, a measure of the substrate concentration at which the reaction rate is half of B underscore max. B. Key kinetic parameters. B underscore max, the maximum rate of reaction, reached when the enzyme is fully saturated with substrate. It depends on the enzyme concentration and is reached when all the enzyme's active sites are occupied by substrate molecules. K underscore M, the substrate concentration at which the reaction rate is half of B underscore max. A low K underscore M indicates high affinity between the enzyme and substrate, while a high K underscore M indicates a lower affinity. Turnover number K underscore cat, the number of substrate molecules converted into product by an enzyme molecule per unit time when the enzyme is fully saturated with substrate. It is a measure of enzyme efficiency. C. Line-Weaver-Burk plot. A Line-Weaver-Burk plot is a double reciprocal graph used to analyze the kinetic data derived from the Michaelis-Menten equation. It plots 1. V1 versus 1. SS1. Giving a straight line with the equation. 1B equals Km V max dot operator 1 S plus 1B max V1 equals V max. Km. Dot operator S 1 plus V max. 1. From the plot, we can determine B underscore max and K underscore M by calculating the slope and intercepts. D. Enzyme inhibition. Enzyme inhibition refers to the decrease in enzyme activity due to the binding of an inhibitor to the enzyme. There are several types of enzyme inhibitors. Competitive inhibition. Inhibitors compete with the substrate for binding to the active site. The inhibition can be overcome by increasing substrate concentration. In competitive inhibition, K underscore M increases, while B underscore max remains unchanged. Non-competitive inhibition. The inhibitor binds to a site other than the active site, causing a conformational change that reduces enzyme activity. In non-competitive inhibition, B underscore max decreases, but K underscore M remains unchanged. In competitive inhibition, the inhibitor binds only to the enzyme substrate complex, preventing the conversion to product. Both B underscore max and K underscore M decrease in incompetitive inhibition. 4. Factors affecting enzyme activity. Several factors influence enzyme activity, including: A. Substrate concentration. The reaction rate increases with increasing substrate concentration until it reaches B underscore max, after which the rate becomes constant. B. Enzyme concentration. An increase in enzyme concentration leads to a higher reaction rate, as more active sites are available for substrate binding. C. Temperature. Enzymes have an optimal temperature range. At higher temperatures, enzyme activity increases until the temperature reaches a point where the enzyme denatures, causing a decrease in activity. D. pH. Each enzyme has an optimal pH at which it is most active. Extreme pH values can alter the enzyme's shape, reducing its activity. E. Presence of activators or inhibitors. Activators increase enzyme activity, while inhibitors decrease it. The type of inhibition: competitive, non-competitive. Incompetitive plays a crucial role in enzyme regulation. 5. Enzyme regulation. Enzyme activity can be regulated in various ways. Allosteric regulation. Some enzymes have allosteric sites, which bind regulatory molecules that affect the enzyme's activity. Positive allosteric effectors increase enzyme activity, while negative allosteric effectors decrease it. Feedback inhibition. In metabolic pathways, the end product often inhibits the enzyme that catalyzes the first step, preventing the overproduction of the product. Covalent modification. Some enzymes are activated or deactivated by covalent modifications, such as phosphorylation. 6. Previous year questions with answers. Question 1, 2016. Which of the following statements is true regarding Michaelis-Menten kinetics? A. The reaction rate is directly proportional to substrate concentration at low strate levels. B. The reaction rate remains constant regardless of concentration. C. The reaction rate decreases with increasing substrate concentration. D. At high substrate concentrations, the reaction rate increases exponentially. Answer. The reaction rate is directly proportional to substrate concentration at low substrate levels. Explanation. At low substrate concentrations, the enzyme is not saturated, so increasing the substrate concentration increases the reaction rate. Question 2, 2018. In competitive inhibition, which of the following is true? A. The inhibitor binds at a site other than the active site. B. The inhibitor increases the value of V_max. C. The inhibitor increases the value of K_m. D. The inhibitor decreases the value of K_m. Answer. C. The inhibitor increases the value of K_m. Explanation. In competitive inhibition, the inhibitor competes with the substrate for the active site, which increases the apparent K_m affinity decreases, but V_max remains unchanged. Question 3, 2020. Which of the following describes enzyme substrate complex formation in enzyme catalysis? A. It decreases the activation energy. B. It increases the activation energy. C. It alters the equilibrium constant of the reaction. D. It prevents the formation of products. Answer. It decreases the activation energy. Explanation. The formation of the enzyme substrate complex lowers the activation energy of the reaction, allowing it to proceed more rapidly. Conclusion. Enzyme kinetics is a crucial area of study in biochemistry and physiology, as it helps to understand how enzymes catalyze reactions and how their activity can be regulated. Through the Michaelis-Menten equation, enzyme activity can be analyzed, and various factors that affect enzyme efficiency, such as substrate concentration, enzyme concentration, temperature, and inhibitors, can be studied. Enzymes are central to the functioning of living organisms, and understanding their kinetics is essential for fields ranging from pharmacology to biotechnology. You said. Enzyme regulation. ChatGPT said. Enzyme regulation. Detailed lecture for CSIR Net JRF Life Sciences. Enzyme regulation is a critical aspect of cellular function. It allows cells to control metabolic processes efficiently, ensuring that enzymes are active only when needed and at appropriate levels. Proper regulation of enzyme activity ensures that the biochemical pathways in a cell are finely tuned to meet the cell's needs, responding to environmental signals, internal metabolic changes, and cellular conditions. Enzyme regulation can occur through several mechanisms, each having significant implications for cellular and organismal functions. 1. Types of enzyme regulation. Enzyme regulation occurs via various mechanisms, including allosteric regulation, feedback inhibition, covalent modification, and the regulation of enzyme synthesis and degradation. A. Allosteric regulation. Allosteric regulation involves the binding of regulatory molecules, effectors or modulators, at sites other than the enzyme's active site, known as allosteric sites. These allosteric sites can be located far from the active site, yet their binding results in a conformational change that affects the enzyme's activity. Allosteric regulation can either activate or inhibit enzyme activity. Allosteric activators. These molecules increase enzyme activity. They bind to the allosteric site and induce a conformational change that enhances the enzyme's ability to bind the substrate. Allosteric inhibitors. These molecules decrease enzyme activity. They bind to the allosteric site and induce a conformational change that reduces the enzyme's affinity for the substrate or makes the enzyme inactive. Allosteric enzymes often exhibit sigmoidal S-shaped curves when the reaction rate is plotted against substrate concentration, as their activity is influenced by both substrate and effector concentrations. Example, the enzyme phosphofructose 1-PFK1 involved in glycolysis is regulated allosterically by ATP, an inhibitor, and AMP, an activator. High ATP levels inhibit PFK1 activity, while low ATP or high AMP activates it. B. Feedback inhibition, end product inhibition. In feedback inhibition, the end product of a metabolic pathway inhibits the activity of an enzyme that catalyzes an early step in the pathway. This is a common method of regulating the flow of metabolites through metabolic pathways, ensuring that the products are not overproduced when they are in sufficient quantities. Mechanism. The end product binds to the enzyme, either at the active site or at an allosteric site, causing a conformational change that reduces the enzyme's activity. This prevents the excessive accumulation of the product. Example, in the biosynthesis of the amino acid isoleucine, isoleucine itself inhibits the enzyme threonine dehydratase, which catalyzes an early step in the pathway, thus preventing the overproduction of isoleucine. C. Covalent modification. Enzymes can be regulated through covalent modification, where chemical groups are added to or removed from the enzyme, leading to changes in its activity. The most common form of covalent modification is phosphorylation, though other modifications like acetylation, methylation, and ubiquitination can also regulate enzyme activity. Phosphorylation. The addition of a phosphate group to specific amino acids such as serine, threonine, or tyrosine can alter the enzyme's conformation, thereby activating or deactivating its function. This modification is catalyzed by kinases, and the phosphate group can be removed by phosphatases. Dephosphorylation. Removal of the phosphate group from an enzyme by phosphatases can lead to the activation or inactivation of the enzyme, depending on the enzyme's nature and the phosphorylation site. Example, the enzyme glycogen phosphorylase is activated by phosphorylation in response to hormones like glucagon or epinephrine, which trigger the breakdown of glycogen. Conversely, the enzyme pyruvate kinase is inactivated by phosphorylation under certain conditions. D. Proteolytic cleavage. Some enzymes are synthesized as inactive precursors, known as cyanogens or coenzymes, and require proteolytic cleavage to become active. This regulation mechanism is important for enzymes involved in processes where premature activation could be harmful, e.g., digestive enzymes or blood clotting factors. Mechanism. A part of the enzyme's polypeptide chain is cleaved to reveal the active site, thus activating the enzyme. Example, the enzyme trypsinogen, the inactive precursor of trypsin, is activated by cleavage of a specific peptide bond in the small intestine. Similarly, prothrombin, the inactive precursor of thrombin, is activated by proteolytic cleavage during blood clotting. E. Regulation of enzyme synthesis and degradation. The amount of an enzyme present in a cell can be regulated by controlling its synthesis or degradation. This process allows the cell to regulate enzyme levels based on its needs. Gene expression. Enzyme levels can be regulated by controlling the transcription of the enzyme's gene. For example, induction occurs when the transcription of a gene is increased in response to certain signals, leading to higher enzyme levels. Repression occurs when the gene's transcription is decreased, reducing enzyme production. Protein degradation. Enzyme activity can be controlled by degradation of the enzyme. The proteasome system and lysosomes are primarily involved in degrading this folder of excess enzymes. Example, the enzyme glucokinase is regulated at the level of gene expression in response to blood glucose levels. When blood glucose is high, the expression of glucokinase is upregulated to promote glucose utilization in the liver. 2. Types of enzyme inhibition. Understanding the mechanisms of enzyme inhibition is crucial for enzyme regulation. The inhibition can be competitive, non-competitive, or incompetitive, each having different effects on enzyme activity and kinetics. Competitive inhibition. Inhibitors bind to the active site of the enzyme, competing with the substrate for binding. This increases the Michaelis constant K_m, but does not affect the V_max. The inhibition can be overcome by increasing the substrate concentration. Non-competitive inhibition. The inhibitor binds to a site other than the active site, causing a conformational change in the enzyme. This type of inhibition decreases the V_max, but does not affect the K_m. In competitive inhibition, the inhibitor binds only to the enzyme substrate complex, preventing the conversion of substrate to product. Both V_max and K_m decrease in incompetitive inhibition. Example, methotrexate, a drug used in cancer therapy, is an example of a competitive inhibitor of the enzyme dihydrofolate reductase. It competes with the substrate dihydrofolate, blocking the enzyme's activity and thus inhibiting DNA synthesis. 3. Previous year questions with answers. Question 1, 2015. Which of the following is true about allosteric enzymes? A. They show a hyperbolic curve in their Michaelis-Menten plot. B. Their activity is regulated by effectors binding to the active site. C. They exhibit cooperative binding, shown by a sigmoidal curve. D. They are not subject to feedback inhibition. Answer. C. They exhibit cooperative binding, shown by a sigmoidal curve. Explanation. Allosteric enzymes exhibit a sigmoidal S-shaped curve in their reaction rate versus substrate concentration plot due to cooperative binding of substrates or effectors. Question 2, 2016. Which of the following types of enzyme inhibition can be overcome by increasing the substrate concentration? A. Competitive inhibition. B. Non-competitive inhibition. C. Incompetitive inhibition. D. Irreversible inhibition. Answer. A competitive inhibition. Explanation. In competitive inhibition, the inhibitor competes with the substrate for the active site, and increasing the substrate concentration can overcome the inhibition. Question 3, 2019. What is the role of covalent modification in enzyme regulation? A. It causes irreversible inhibition of enzyme activity. B. It involves the addition or removal of phosphate groups to regulate enzyme function. C. It increases the enzyme's affinity for the substrate. D. It regulates the enzyme through allosteric sites. Answer. B. It involves the addition or removal of phosphate groups to regulate enzyme function. Explanation. Covalent modification, such as phosphorylation and dephosphorylation, is a key mechanism in enzyme regulation, allowing rapid activation or deactivation in response to signals. Conclusion. Enzyme regulation is essential for controlling metabolic pathways, ensuring that the cell's resources are used efficiently and that cellular processes occur only when needed. Through mechanisms like allosteric regulation, feedback inhibition, covalent modification, and regulation of enzyme synthesis and degradation, cells maintain precise control over enzyme activity. Understanding these processes is crucial for interpreting how metabolic pathways are coordinated and how drugs and other molecules can modulate enzyme function in therapeutic contexts. You said. Mechanism of enzyme catalysis. ChatGPT said. Mechanism of enzyme catalysis. Detailed lecture for CSIR.net JRF Life Sciences. Enzyme catalysis is a fundamental process that accelerates the rate of biochemical reactions, enabling essential metabolic processes to occur at a pace compatible with life. Enzymes are biological catalysts, typically proteins, that lower the activation energy of a reaction, allowing it to proceed much faster than it would without the enzyme. The efficiency of enzyme catalysis lies in their ability to stabilize the transition state of the reaction and bring substrates into optimal orientation, minimizing the energy required to reach the transition state. The mechanism of enzyme catalysis involves several key steps that facilitate the conversion of substrates into products by decreasing the energy barrier for the reaction. This can occur through various modes of action, including acid-base catalysis, covalent catalysis, metal ion catalysis, and proximity effects. The mechanism of enzyme catalysis can be understood through a series of steps and concepts as described below. 1. Steps in enzyme catalysis. Enzyme catalysis can be broken down into several stages, each of which contributes to the efficient transformation of substrate into product. A. Substrate binding to the active site. The first step in enzyme catalysis involves the substrates binding to the enzyme's active site, a specialized region of the enzyme with a unique three-dimensional structure. The active site is highly specific for its substrates, and the binding is facilitated by non-covalent interactions such as hydrogen bonds, van der Waals forces, and electrostatic interactions. The enzyme substrate complex, S complex, is formed through these interactions. Induced fit model. In this model, the enzyme undergoes a conformational change upon binding with the substrate, optimizing the interaction between the enzyme and the substrate. This model suggests that the enzyme's active site is flexible and adapts to the substrate shape. Lock and key model. This model proposes that the enzyme's active site is already complementary to the substrate shape, and the substrate fits perfectly into the active site like a key fitting into a lock. B. Formation of the transition state. Once the substrate binds to the active site, the enzyme stabilizes the transition state of the reaction. The transition state is a high-energy intermediate state in which bonds are partially formed and broken as the substrate is converted into the product. Transition state stabilization. Enzymes lower the activation energy by stabilizing the transition state. This can be achieved by providing an environment in the active site that facilitates the formation of the transition state, such as through precise positioning of catalytic residues, altering the substrate's geometry, or providing a favorable microenvironment. C. Catalysis. Mechanisms involved. Enzymes use a variety of mechanisms to catalyze reactions, each of which helps to stabilize the transition state and lower the activation energy. Some of the key mechanisms include acid-base catalysis. In this mechanism, the enzyme donates or accepts protons H during the reaction. Acid-base catalysis can occur when amino acid residues in the enzyme's active site have functional groups capable of acting as acids or bases such as histidine, aspartic acid, or glutamic acid. These groups either donate a proton to the substrate or accept a proton from it, facilitating bond breakage or formation. Example: in the hydrolysis of peptide bonds by chymotrypsin, the enzyme uses a histidine residue to act as a general base that abstracts a proton from a water molecule, enhancing its nucleophilicity. Covalent catalysis. In covalent catalysis, the enzyme forms a transient covalent bond with the substrate during the reaction. This bond formation alters the reaction pathway, creating an intermediate that is more reactive than the original substrate. The enzyme's covalent bond with the substrate is eventually broken, releasing the product. Example: in the case of serine proteases, e.g., the enzyme's covalent bond with the substrate is eventually broken, releasing the product. Example: in the case of serine proteases, e.g., chymotrypsin, the serine residue in the enzyme's active site forms a covalent bond with the substrate, facilitating the breaking of peptide bonds. Metal ion catalysis. Some enzymes require metal ions as cofactors for their catalytic activity. These metal ions participate in catalysis by stabilizing negative charges on the substrate or transition state, activating substrates, or facilitating electron transfer during the reaction. Example: carbonic anhydrase, which contains a zinc ion in its active site, uses the metal ion to activate water molecules, enabling the conversion of carbon dioxide and water into bicarbonate and protons. Proximity and orientation effects. Enzymes increase the rate of reaction by bringing the substrates into close proximity and in the correct orientation for the reaction to occur. This minimizes the entropy of the system and ensures that the molecules collide with the proper orientation for the reaction to proceed. Example: in the enzyme acetylcholinesterase, the active site brings the substrate acetylcholine into close proximity with catalytic residues, facilitating the hydrolysis of acetylcholine into acetic acid and choline. Strain or distortion mechanism. Enzymes can also induce strain or distortion in the substrate, making it more reactive. By binding to the substrate in a way that distorts its structure, the enzyme lowers the energy required to reach the transition state. Example: in lysozyme, the enzyme binds to the sugar substrate in a way that distorts its structure, facilitating the breaking of the glycosidic bond. D. Product formation and release. Once the transition state has been stabilized and the reaction has proceeded to the products, the enzyme facilitates the breakdown of the transition state, and the product is formed. The enzyme product complex EP is then formed, which eventually dissociates into free enzyme and product, ready to catalyze another round of the reaction. Product release. The enzyme has a lower affinity for the product than for the substrate, which ensures the release of the product and the return of the enzyme to its original state. 2. Key concepts in enzyme catalysis. Understanding enzyme catalysis involves not only the mechanisms of action but also kinetic principles that govern enzyme efficiency. Michaelis-Menten kinetics. The relationship between the reaction rate and substrate concentration is described by the Michaelis-Menten equation, where the rate of reaction V increases with increasing substrate concentration until it reaches the maximum velocity V_max, at which point the enzyme is saturated with substrate. The Michaelis constant K_m reflects the affinity of the enzyme for the substrate. Enzyme inhibition. Enzyme activity can be modulated by inhibitors, which can be competitive, non-competitive, or incompetitive. Inhibition can affect enzyme catalysis by altering the enzyme's ability to bind to the substrate or catalyze the reaction. Turnover number K_cat. The turnover number represents the number of substrate molecules converted to product per enzyme molecule per unit of time under optimal conditions. This value provides an estimate of the catalytic efficiency of an enzyme. 3. Previous year questions with answers. Question 1, 2017. Which of the following is not a mechanism of enzyme catalysis? A. Acid-base catalysis. B. Covalent catalysis. C. Polarization of the enzyme active site. D. Strain distortion mechanism. Answer. C. Polarization of the enzyme active site. Explanation. While enzymes stabilize the transition state and use mechanisms like acid-base catalysis, covalent catalysis, and strain distortion, polarization of the active site is not recognized as a direct mechanism of enzyme catalysis. Question 2, 2018. Which of the following enzymes uses metal ion catalysis in its mechanism? A. Lysozyme. B. Chymotrypsin. C. Carbonic anhydrase. D. Hexokinase. Answer. C. Carbonic anhydrase. Explanation. Carbonic anhydrase uses a zinc ion to facilitate the hydration of carbon dioxide to bicarbonate and protons. Question 3, 2020. In enzyme catalysis, the lowering of activation energy by the enzyme is most closely associated with a. The enzyme stabilization of the substrate. B. The enzyme stabilization of the transition state. C. The enzyme stabilization of the product. D. The increase in substrate concentration. Answer. B. The enzyme stabilization of the transition state. Explanation. Enzymes lower activation energy primarily by stabilizing the transition state, making it easier for the reaction to proceed. Conclusion. Enzyme catalysis is a sophisticated and highly efficient mechanism that is essential for life. Enzymes accelerate biochemical reactions by lowering the activation energy required for the reaction to proceed, achieved through various catalytic mechanisms such as acid-base catalysis, covalent catalysis, metal ion catalysis, proximity effects, and strain distortion. Understanding these processes is fundamental to comprehending how enzymes function in cellular processes and how they can be manipulated for therapeutic purposes. You said. Isozymes. ChatGPT said. Isozymes. Detailed lecture for CSIR.net JRF Life Sciences. Isozymes, also known as isoenzymes, are different molecular forms of an enzyme that catalyze the same reaction but may differ in their physical properties, such as structure, kinetic behavior, and regulatory mechanisms. These enzymes arise from the expression of different gene variants or alleles, which may encode slightly different versions of the same enzyme. Isozymes serve to fine-tune biochemical processes, allowing organisms to adapt to various environmental conditions or developmental stages. 1. Definition and overview. Isozymes are distinct enzyme variants that catalyze identical biochemical reactions but exhibit differences in their physical characteristics, including their amino acid sequence, electrophoretic mobility, kinetic properties, and sometimes even their substrate specificity. The presence of isozymes in different tissues, developmental stages, or under various environmental conditions allows for regulation of metabolic pathways in a more precise manner. These enzymes may also differ in optimal temperature, pH, and activation energy, enabling organisms to adjust their metabolism according to varying physiological demands. Isozymes are generally produced by different gene loci or allelic variants of a single gene, meaning they may have subtle differences in structure but catalyze the same reaction. 2. Mechanism of isozyme formation. Isozymes are formed due to genetic variations at different loci, alternative splicing of mRNA, or post-translational modifications. These variations lead to slightly different structures that affect the enzyme's substrate affinity, catalytic efficiency, and regulatory properties. Gene duplication. A common method of isozyme formation is through gene duplication. Over evolutionary time, a duplicated gene may accumulate mutations that create a new form of the enzyme with altered properties, but still capable of catalyzing the same reaction. Alternative splicing. This process generates multiple enzyme forms from a single gene by splicing the mRNA in different ways. This mechanism allows for the production of tissue-specific isozymes or forms that are active under different environmental conditions. 3. Properties and significance of isozymes. Isozymes exhibit a variety of properties that are important for the regulation of metabolic processes. Some of the key features include differential tissue distribution. Different tissues in the body may express distinct isozymes of the same enzyme. For example, lactate dehydrogenase (LDH) has multiple isozymes, and different tissue types, muscle, liver, heart may express different forms with different kinetic properties suited to their metabolic needs. Developmental regulation. Isozyme expression is often regulated during development. For instance, an embryo may use one isozyme of a particular enzyme, which is replaced by a different isozyme in the adult stage. Environmental adaptation. Isozymes allow organisms to adapt to environmental changes, such as temperature fluctuations or oxygen availability. For example, catalase isozymes. 3. Previous year questions with answers. Question 1, 2017. Which of the following is not a mechanism of enzyme catalysis? A. Acid-base catalysis. B. Covalent catalysis. C. Polarization of the enzyme active site. D. Strain distortion mechanism. Answer. C. Polarization of the enzyme active site. Explanation. While enzymes stabilize the transition state and use mechanisms like acid-base catalysis, covalent catalysis, and strain distortion, polarization of the active site is not recognized as a direct mechanism of enzyme catalysis. Question 2, 2018. Which of the following enzymes uses metal ion catalysis in its mechanism? A. Lysozyme. B. Chymotrypsin. C. Carbonic anhydrase. D. Hexokinase. Answer. C. Carbonic anhydrase. Explanation. Carbonic anhydrase uses a zinc ion to facilitate the hydration of carbon dioxide to bicarbonate in protons. Question 3, 2020. In enzyme catalysis, the lowering of activation energy by the enzyme is most closely associated with a. The enzyme stabilization of the substrate. B. The enzyme stabilization of the transition state. C. The enzyme stabilization of the product. D. The increase in substrate concentration. Answer. B. The enzyme stabilization of the transition state. Explanation. Enzymes lower activation energy primarily by stabilizing the transition state, making it easier for the reaction to proceed. Conclusion. Enzyme catalysis is a sophisticated and highly efficient mechanism that is essential for life. Enzymes accelerate biochemical reactions by lowering the activation energy required for the reaction to proceed, achieved through various catalytic mechanisms such as acid-base catalysis, covalent catalysis, metal ion catalysis, proximity effects, and strain distortion. Understanding these processes is fundamental to comprehending how enzymes function in cellular processes and how they can be manipulated for therapeutic purposes. You said. Isozymes. ChatGPT said. Isozymes. Detailed lecture for CSIR Net JRF Life Sciences. Isozymes, also known as isoenzymes, are different molecular forms of an enzyme that catalyze the same reaction but may differ in their physical properties, such as structure, kinetic behavior, and regulatory mechanisms. These enzymes arise from the expression of different gene variants or alleles, which may encode slightly different versions of the same enzyme. Isozymes serve to fine-tune biochemical processes, allowing organisms to adapt to various environmental conditions or developmental stages. 1. Definition and overview. Isozymes are distinct enzyme variants that catalyze identical biochemical reactions but exhibit differences in their physical characteristics, including their amino acid sequence, electrophoretic mobility, kinetic properties, and sometimes even their substrate specificity. The presence of isozymes in different tissues, developmental stages, or under various environmental conditions allows for regulation of metabolic pathways in a more precise manner. These enzymes may also differ in optimal temperature, pH, and activation energy, enabling organisms to adjust their metabolism according to varying physiological demands. Isozymes are generally produced by different gene loci or allelic variants of a single gene, meaning they may have subtle differences in structure. Explanation. While enzymes stabilize the transition state and use mechanisms like acid-base catalysis, covalent catalysis, and strain distortion, polarization of the active site is not recognized as a direct mechanism of enzyme catalysis. Question 2, 2018. Which of the following enzymes uses metal ion catalysis in its mechanism? A. Lysozyme. B. Chymotrypsin. C. Carbonic anhydrase. D. Hexokinase. Answer. C. Carbonic anhydrase. Explanation. Carbonic anhydrase uses a zinc ion to facilitate the hydration of carbon dioxide to bicarbonate in protons. Question 3, 2020. In enzyme catalysis, the lowering of activation energy by the enzyme is most closely associated with a. The enzyme stabilization of the substrate. B. The enzyme stabilization of the transition state. C. The enzyme stabilization of the product. D. The increase in substrate concentration. Answer. B. The enzyme stabilization of the transition state. Explanation. Enzymes lower activation energy primarily by stabilizing the transition state, making it easier for the reaction to proceed. Conclusion. Enzyme catalysis is a sophisticated and highly efficient mechanism that is essential for life. Enzymes accelerate biochemical reactions by lowering the activation energy required for the reaction to proceed, achieved through various catalytic mechanisms such as acid-base catalysis, covalent catalysis, metal ion catalysis, proximity effects, and strain distortion. Understanding these processes is fundamental to comprehending how enzymes function in cellular processes and how they can be manipulated for therapeutic purposes. You said. Isozymes. ChatGPT said. Isozymes. Detailed lecture for CSIR Net JRF Life Sciences. Isozymes, also known as isoenzymes, are different molecular forms of an enzyme that catalyze the same reaction but may differ in their physical properties, such as structure, kinetic behavior, and regulatory mechanisms. These enzymes arise from the expression of different gene variants or alleles, which may encode slightly different versions of the same enzyme. Isozymes serve to fine-tune biochemical processes, allowing organisms to adapt to various environmental conditions or developmental stages. 1. Definition and overview. Isozymes are distinct enzyme variants that catalyze identical biochemical reactions but exhibit differences in their physical characteristics, including their amino acid sequence, electrophoretic mobility, kinetic properties, and sometimes even their substrate specificity. The presence of isozymes in different tissues, developmental stages, or under various environmental conditions allows for regulation of metabolic pathways in a more precise manner. These enzymes may also differ in optimal temperature, pH, and activation energy, enabling organisms to adjust their metabolism according to varying physiological demands. Isozymes are generally produced by different gene loci or allelic variants of a single gene, meaning they may have subtle differences in structure but catalyze the same reaction. 2. Mechanism of isozyme formation. Isozymes are formed due to genetic variations at different loci, alternative splicing of mRNA, or post-translational modifications. These variations lead to slightly different structures that affect the enzyme's substrate affinity, catalytic efficiency, and regulatory properties. Gene duplication. A common method of isozyme formation is through gene duplication. Over evolutionary time, a duplicated gene may accumulate mutations that create a new form of the enzyme with altered properties, but still capable of catalyzing the same reaction. Alternative splicing. This process generates multiple enzyme forms from a single gene by splicing the mRNA in different ways. This mechanism allows for the production of tissue-specific isozymes or forms that are active under different environmental conditions. 3. Properties and significance of isozymes. Isozymes exhibit a variety of properties that are important for the regulation of metabolic processes. Some of the key features include differential tissue distribution. Different tissues in the body may express distinct isozymes of the same enzyme. For example, lactate dehydrogenase (LDH) has multiple isozymes, and different tissue types, muscle, liver, heart, may express different forms with different kinetic properties suited to their metabolic needs. Developmental regulation. Isozyme expression is often regulated during development. For instance, an embryo may use one isozyme of a particular enzyme, which is replaced by a different isozyme in the adult stage. Environmental adaptation. Isozymes allow organisms to adapt to environmental changes, such as temperature fluctuations or oxygen availability. For example, catalase isozymes in various species may differ in their sensitivity to temperature or pH, allowing them to better cope with oxidative stress under different conditions. Regulation of metabolic pathways. Isozymes can have different Michaelis constants K_m and maximum velocities V_max, which can provide nuanced control over metabolic pathways. This allows the body to regulate the rate of a biochemical reaction more efficiently depending on the specific demands of the cell or tissue. 4. Examples of isozymes. Some well-known examples of isozymes include lactate dehydrogenase (LDH). LDH catalyzes the conversion of pyruvate to lactate and vice versa. It has 5 isozymes LDH1 to LDH5 that differ in tissue distribution and kinetic properties. For example, LDH1 is predominantly found in the heart and red blood cells, where it has a high affinity for lactate. LDH5 is found in skeletal muscles and has a lower affinity for lactate, supporting anaerobic conditions. Creatine kinase (CK). Creatine kinase has 3 isozymes CKM, CKMB, CKBB that are found in skeletal muscle, heart muscle, and brain, respectively. These isozymes catalyze the reversible phosphorylation of creatine, which is important for energy storage and transfer. Aconitase. Aconitase, an enzyme involved in the citric acid cycle, has isoforms that can differ in their sensitivity to regulatory molecules such as iron. These isozymes are present in different tissues, adapting to varying metabolic needs. Alkaline phosphatase. Alkaline phosphatase exists as several lysozymes found in different tissues such as liver, bone, and intestine. These isozymes have varying substrate specificities, which allows for the regulation of phosphate metabolism in distinct physiological contexts. Glucose 6-phosphate dehydrogenase (G6PD). G6PD is essential for maintaining the NADPH pool and protecting cells against oxidative damage. Isozymes of G6PD are found in various tissues, with distinct activity levels depending on the tissue's oxidative stress response. 5. Importance of isozymes in clinical diagnosis. Isozymes are not only important for understanding metabolic regulation but also serve as valuable biomarkers for diagnostic purposes. For example, lactate dehydrogenase (LDH) levels are measured in patients with heart attacks or muscle injury, as different LDH isozymes are released into the bloodstream during tissue damage. High levels of LDH1 in the serum suggest myocardial infarction, while elevated levels of LDH5 indicate muscle damage. Creatine kinase (CK) is used to assess muscle damage or heart attacks. Elevated CKMB levels are indicative of myocardial infarction. Alkaline phosphatase (ALP) levels can help diagnose bone diseases, e.g., Paget's disease or liver dysfunction, as different isoforms are secreted by the liver, bone, and intestine. 6. Previous year questions with answers. Question 1, 2016. Which of the following statements about isozymes is false? A. Isozymes catalyze the same reaction. B. Isozymes have the same amino acid sequence. C. Isozymes can differ in their regulation. D. Isozymes may exhibit different kinetic properties. Answer. B. Isozymes have the same amino acid sequence. Explanation. Isozymes catalyze the same reaction, but they may have different amino acid sequences, which results in differences in their kinetic properties and regulatory mechanisms. Question 2, 2018. Which of the following is not an example of an isozyme? A. Lactate dehydrogenase (LDH). B. Creatine kinase (CK). C. Alcohol dehydrogenase (ADH). D. Pepsinogen. Answer. D. Pepsinogen. Explanation. Pepsinogen is a precursor of pepsin and is not an isozyme. LDH, CK, and ADH all have isoenzymes that are present in different tissues and have different properties. Question 3, 2019. Which of the following is an example of an isozyme that exhibits tissue-specific distribution? A. Lactate dehydrogenase (LDH). B. Hexokinase. C. Glucokinase D. All of the above. Answer. A. Lactate dehydrogenase (LDH). Explanation. LDH has different isozymes LDH1 to LDH5 that are expressed in different tissues, such as heart, muscle, and liver, and these isozymes differ in their kinetic properties. Conclusion. Isozymes are essential for the regulation and fine-tuning of metabolic processes in different tissues, under varying physiological conditions, and during developmental stages. These enzyme variants provide a level of control that cannot be achieved by a single enzyme, allowing organisms to These enzyme variants provide a level of control that cannot be achieved by a single enzyme, allowing organisms Isozymes are essential for the regulation and fine-tuning of metabolic processes in different tissues, under varying physiological conditions, and during developmental stages. These enzyme variants provide a level of control that cannot be achieved by a single enzyme, allowing organisms to optimize metabolic efficiency and adaptability. Understanding the properties, distribution, and role of isozymes is crucial for both basic biology and clinical diagnostics, as their dysfunction can be indicative of various diseases. You said. Confirmation of proteins. ChatGPT said. Confirmation of proteins. CSIRNet JRF Life Sciences Lecture. Ultra detailed. Paragraph-based theory with PYQs. This is your complete, in-depth coaching material. No other notes required. Proteins are fundamental biomolecules responsible for the majority of structural and functional roles in biological systems. Their function is entirely dependent on their three-dimensional conformation, which results from the specific sequence of amino acids' primary structure folding into defined shapes through various levels of structural organization. The term conformation of proteins refers to the spatial arrangement of atoms in a protein molecule, particularly the folding patterns of the polypeptide backbone and the side chains of amino acid residues. These conformations are dynamic and stabilized by various non-covalent interactions. Understanding protein conformation is crucial for interpreting enzyme activity, signaling pathways, immune responses, and molecular interactions, all of which are highly tested in the CSIRNet Life Sciences exam. 1. Primary, secondary, tertiary, and quaternary structures. Primary structure refers to the linear sequence of amino acids linked by peptide bonds. This linear sequence determines all higher levels of protein structure. Secondary structure involves regular, recurring arrangements of the polypeptide chain, stabilized primarily by hydrogen bonds. The two major types are alpha-alpha helix, a right-handed coil stabilized by hydrogen bonds between the cardinal oxygen of residue I and the amide hydrogen of residue I+4. Common in fibrous and globular proteins. Beta-beta pleated sheet, formed by beta strands linked laterally by at least 2 or 3 backbone hydrogen bonds, can be parallel or antiparallel. Turns and loops, non-repetitive regions that connect helices and sheets, often found on the protein surface. Tertiary structure represents the overall 3D conformation of a single polypeptide chain. It includes